We focused on the cytoprotective and reparative effect of petadecapeptide BPC-157 on blood, liver and kidney cells after cisplatin-induced DNA damage. First, BPC-157 is a very safe and stable anti-ulcer peptide successfully tested in phase 2 clinical trials on IBD patients. It also portrayed an ability to heal fistulas. Subsequently, besides gastrointestinal lesions, BPC-157 markedly counteracted various liver lesions. Therefore, BPC-157 as a stable anti-ulcer peptide, stable in human gastric juice and thereby easily applicable, safe, inexpensive, performs very important functions in the gastrointestinal tract, and is suggested to be an antagonist of non-steroid anti-inflammatory drug toxicity. Therefore, our next focus was protective and reparative effect of petadecapeptide BPC-157 on blood, liver and kidney cells in mice. We used the regular COMET test and the micronucleus test to demonstrate the effect on DNA damage induced by a noxious agent, in this case a cytostatic agent such as cisplatin. The above protocol was used. BPC-157 counteracts the damaging effect of cisplatin in Swiss albino mice lymphocytes. The joint administration of BPC-157 and cisplatin resulted in less damage incurred by cisplatin. In the table, we can see that lymphocyte damage after 1 hour was 2.9% versus cisplatin of 24.4%. Kidney damage was 0.3% versus 14.2% of cisplatin. Liver damage was 2.8% versus 12.2% in cisplatin. BPC-157 counteracts the damaging effect of cisplatin in Swiss albino mice kidney cells. BPC-157 counteracts the damaging effect of cisplatin in Swiss albino mice liver cells. BPC-157 counteracts the damaging effect of cisplatin in Swiss albino mice reticulocytes. This was shown by the micronucleus test. The micronuclei values in BPC-157 are equal to the control group. It has been demonstrated that the joint effect of BPC and cisplatin decreases the micronuclei count after 24 hours. The most sensitive organ to the toxic effects of cisplatin was the kidney, followed by the liver and lymphocytes. Therefore, whatever the effect of cisplatin, BPC-157 may fully counteract its damaging effect. In conclusion, the results indicated that BPC-157 has a protective effect on healthy cells. A protective effect of BPC-157 is most likely based on a reduced intake of cisplatin into the cell or its participation in the process of DNA reparation of damaged cells. This requires further research.